This presentation covers work to date on Certip project ER-2419, an effort to develop a high-resolution passive profiler to aid contaminated groundwater investigations. The overall goal of this research was to develop and demonstrate a delineation tool for the saturated subsurface at high resolution. Specifically, we were to directly measure groundwater and contaminant flux at the centimeter scale, be able to quantify the biogeochemical conditions at the same scale, assess microbial community structure and activity using compound-specific isotope analysis of the chlorinated solvents, and determine the extent to which the high-resolution data could be used to improve site conceptual models. The driver for this work was the current problems in understanding contaminated sites that have heterogeneous stratigraphy. This complicates the analysis of what is occurring in the subsurface in a number of ways, one of which is understanding groundwater velocity. Low permeable regions, clay lenses, silty sands layered within a more permeable sand will cause differential velocity and can lead to misunderstandings of contaminant fate due to the fact that the well screens typically pull from the most permeable layers, and oftentimes at sites the well screens are fairly large, 5 feet, 10 feet, crossing multiple zones and leading to a oversimplification of the groundwater velocity profiles. In addition, the heterogeneous stratigraphy will often lead to high concentration zones in low permeability regions of the chlorinated solvents. And again, well screens that pull across multiple zones typically pull water from the most permeable zone, in which case areas of high concentration or even DNAPL will be missed. This can cause issues with site rebound or longer cleanup periods than expected due to the fact that the mass of solvent in the low permeable regions is not accurately described. It's also important if an accurate site assessment for what is occurring in the contaminated area in terms of microbial or abiotic processes is the need to have high resolution data on which terminal electron accepting process is dominating at each zone. This can allow better site decisions to be made on whether abiotic or biotic remediation efforts will take place and where the amendments should be placed. So how would higher resolution data potentially impact the site assessment or remedial activities? Well, higher resolution data, particularly more accurate information, would allow you to better model contaminant transport, particularly with respect to low permeability zones, in which case wells often have difficult times returning accurate information. It could help determine realistic cleanup goals or duration of cleanup, allow you to better predict if there's going to be rebound due to dissolution of trapped DNAPL, and also potentially allow you to determine the transport time out of the low permeability zones. It could aid in the selection of remedial processes, so whether it's better to go with a biological, bioaugmentation, biostimulation, or chemical process such as nano iron or other physical processes such as heating. It can also help with the location and frequency of application to better be able to pinpoint where the amendments will have the greatest effect. The basic technology that we are using to develop the sampler is based on equilibrium pore water sampling. This technology has been around for over 50 years and essentially works by allowing a volume of water within the sampler which is separated from the surrounding sediment by a porous membrane, usually 0.2 to 0.45 micron, to equilibrate with the sediment water over time. So once the sampler is in place, the concentration of the water starts off at 0, and over time the dissolved constituents in the pore water will diffuse through the membrane into the cell, eventually equilibrating after a given amount of time. The time is variable. It depends on the specifications of the sampler, but generally is somewhere between 7 and 21 days, depending on the specific design of interest. Any dissolved species can be evaluated in this manner. In general, the only limitation is the volume of water required for the analysis. The technology has been used to evaluate quite a large number of dissolved species in shallow sediments. These include chloride, bromide, nitrate, ammonium, dissolved organic carbon, sulfate, various contaminants, RDX, perchlorate, BTEX, chlorinated solvents, TCE, PC, TECA, as well as numerous metals have also been evaluated. It's typically been used in shallow environments, wetlands, rivers and lake sediments, and shallow aquifers, but there have been some applications to deeper sediments and shallow groundwaters. The advantages are that it has the least sediment disturbance, so it's more likely to return what's actually occurring at that site. It's very simple to use. It can provide very high spatial resolution. It can go down to centimeter scale resolution. It's low cost. It's essentially the cost of insertion and retrieval plus analysis. It avoids the cost of well installation or the issue of well installation in a location which is not useful. And it also allows you to calculate concentrations on ore fluxes. It's been used to evaluate a number of processes, fate of contaminants, bioavailability, monitoring remediation effectiveness, and plant contaminant interactions. And its really only disadvantage is that because the amount of water per depth is limited, the detection limit is often not as low as for well sampling. But generally, you're only talking about a factor of 5 or 10 greater detection limits than a typical method. One of our objectives in developing the sampler was to extend the capabilities of the traditional equilibrium pore water samplers. And an important analysis that can greatly aid site evaluations is the knowledge of the overall microbial community and activity of that community at the site. Typically this is done by placing a biotrap in a well and then analyzing the bacteria that are captured on the biotrap. But again, the well is going to return information on the most permeable region, and it can be very difficult to get sufficient water in very low permeable regions. So what we did was we essentially took the same technology as the biotrap technology. We used the same bioset beads, but instead of placing them in the well, we placed them in the sampler. In the middle of the slide there's a photograph of a prototype sampler, and the red box surrounds a simple cell in which bioset beads can be placed. There's a stainless steel mesh that's placed over the beads to protect them from the during insertion, and also allow pore water to reach the beads. The beads equilibrate with the bacterial community. Bacteria colonize the beads, and after a given amount of time, 2 to 3 weeks, the sampler can be removed, the beads are removed, and shipped to commercial labs for analysis. And there's a very wide range of analyses that are possible. Molecular techniques can be used to evaluate the types of reductive dechlorinators, the relative numbers of dechlorinators, the types of bacteria that perform specific processes such as denitrification or sulfate reduction, or even have the capacity for certain co-metabolic processes. And so that's a very powerful technique to be able to look at what the microbial community, how it varies with depth. In this case, the sampler can evaluate every 20 centimeters. But beyond this, the same technology can also be used to evaluate the activity of that population. And to do this, use a technique called compound-specific isotope analysis, which we will discuss next. One of the more powerful techniques for evaluating what's occurring at a site is compound-specific isotope analysis. This analysis works by evaluating the ratio of carbon-13 to carbon-12 when bacteria degrade chlorinated solvents. They fractionate it, which means that the parent compound, in the case of this picture, trichloroethylene, becomes enriched in C13, and the daughter product, cis-2Ce, becomes lighter, becomes depleted in C13. And so the degree of fractionation is relative to the amount of degradation that occurs. So if you have TCE that is much heavier than the parent, or rather its original source, or cis, which is very light, you can determine in a relative manner whether or not degradation is occurring at that site. The bioset beads that are used for microbial analysis can also be used to evaluate the isotopic composition of chlorinated solvents as well. So the beads can be shipped to a commercial lab, they desorb the chlorinated solvents, and they can determine the stable isotopic composition of the chlorinated solvents. And so what this allows us to do is to evaluate the chlorinated solvents with depth to determine whether they're being degraded at high resolution, around every 20 centimeters, as well as what bacteria are present. And so combining all these together gives you much more information about what occurs at each individual depth than you would otherwise be able to get unless you put in very, very high resolution wells, well screens every 1 foot. And even then, it's possible that if it was a low permeability region, some of the well screens would not return data because the sediment around it would be have too low a flow rate to be able to sample by well. Another very important piece of information to be able to understand what's going on at a contaminated groundwater site is pore velocity. Pore velocity is obviously a critical parameter in terms of modeling what's going to happen at the site, where the contaminants are moving, how long it will take for them to move out of low permeable regions into high permeable regions, as well as dissolution rates. And so we wanted to modify the traditional pore water equilibrium sampler to be able to determine pore velocity. The way we did this, or the approach that we're using, is to be able to correlate a mass transfer coefficient of a conservative tracer to pore velocity. So in this case we're using bromide, so the cells in the sampler start off with a given concentration of bromide, typically 100 milligrams per liter, which is usually well above background unless you're in a brackish situation. We use cells of different specific surface area, so the cells have different volume to area ratios. And what that produces is different rates of equilibrium. So the more surface area per volume is, the faster the conservative tracer is going to diffuse out of the cell and into the pore water. The lower the specific surface area, the slower it equilibrates. And so if for one time point, by using 4 different specific surface areas, we can determine the mass transfer coefficient with one deployment. And the equation in the graph is the equation that describes the equilibration of the conservative tracer with time as a function of the mass transfer coefficient and the specific surface area. So if we're able to determine the mass transfer coefficient for a given depth, we can then use a relationship between the magnitude of the mass transfer coefficient and the velocity. The mass transfer coefficient should increase proportionally with velocity for a number of reasons, which we can we'll discuss in a slide or two. This slide is just an overview of how we do the experiments to correlate the mass transfer coefficient with the velocity. So we have a flow cell. That flow cell is about 2 feet by 2 feet, and it's about a foot wide. You can see in the picture that there are sample ports in the midline of the flow cell, and there's a prototype of the sampler propped against the box in order to show what it looks like. There are 3 sets of cells on this sampler, and so we return 3 data points every time we insert the sampler. And you can see that the 4 different sample cells vary in specific surface area based on the depth of the cell as well as the opening of the cell. So we fill the cells with a bromide solution. We insert the sampler into the sand box. We equilibrate for a given amount of time. We remove the sampler and measure how much of the bromide has been lost from the cell. And then we can do that for various velocities. So we can look at 0 centimeters a day, 1 centimeter a day, up to 100 centimeters a day, or any other velocity we're interested in. So to date, we've evaluated a number of different velocities. In the bottom right-hand corner of the slide, there's a plot of the pore velocity in centimeters a day versus the measured mass transfer coefficient. And as we expected, the mass transfer coefficient increases with increasing velocity. It looks like in a non-linear manner, and so the mass transfer coefficient will eventually reach a maximum. Now, if the velocity is over 100 centimeters a day, you're in a very permeable zone, and so this technique might not be as useful as typical wells would be able to return the information that's required. But as you move to the left, down to low velocities, 1 centimeter a day, 5 centimeter a day, those are very small velocities and would be of great use for site evaluation to evaluate low permeability regions. The figure in the upper left-hand corner simply demonstrates that the mass transfer coefficient is independent of the deployment time. So regardless of how long you deploy the sampler, as long as the sampler hasn't come to equilibrium with the pore water, the mass transfer coefficient is not varying, which means that we have a wide latitude in the amount of time that the sampler can be deployed. So what we think we've demonstrated is the ability of the sampler to evaluate pore velocity with a single time deployment with a fairly robust relationship between velocity and the mass transfer coefficient. In order to better understand the observed relationship between the mass transfer coefficient and the velocity in the pore water, we've developed a analytical model with, in collaboration with Dr. Danny Reibel, to look theoretically at the expected relationship between pore velocity and the mass transfer coefficient. So the model is a 2D quasi-steady state model which relates the diffusive flux out of the cells with the dispersive flux away from the cell. Its parameters are parameters that would not be available in the field, but we can use the theoretical relationship and known parameters in the lab to evaluate the relationship that we see. And so the solution to the problem results in an equation in which the mass transfer coefficient Km is a function of a number of constants and, most importantly, obviously, the pore water velocity. And so the parameters are all defined on the slide, and obviously some of the key parameters are the tortuosity, the porosity, the diffusion coefficient, the resistance to the membrane. But most importantly for us is the fact that the mass transfer coefficient is proportional to the velocity. So evaluating the model for expected values of the various constants, the diffusion coefficient of bromide and water, the dispersion coefficient, the tortuosity of the sediment based on the porosity, we had plotted the theoretical curve, which is the solid line in the large figure, with the experimental data. And the model appears to fit the data quite well, giving us further confidence that the relationship we see is not only a rigorous relationship in terms of multiple data points for each velocity returning a strong correlation, but that also the theoretical relationship conforms to the data we see, and that the change in velocity produces a change in the mass transfer as expected. So we developed a prototype sampler about a year ago. The sampler was based on structural analysis as well as the requirements for each type of data point we were trying to return. And so the sampler, the first prototype sampler, is a 2-inch or 5-centimeter diameter 316 stainless steel rod, in which every 20 centimeters we have full data sets, which include the measurement for velocity, the measurement of chlorinated solvents, the measurement of dissolved species for geochemical analysis, so nitrate, nitrite, total iron, sulfate, chloride, bromide, anything else you would like to measure, and then as well the bioset beads for microbial diversity populations and the compound-specific isotope analysis of the chlorinated solvents. The device is made to be directly coupled to a standard geoprobe drive rod for direct insertion, and the cells are separated from the sediment by a series of materials to both protect the cells and also provide a diffusive a diffusive link to the sediment. So there's a stainless steel coarse mesh that goes at the top to protect the cells from the sediment during insertion. There's a Viton membrane which does not cover the cells, but is used as a seal to prevent sediment from migrating around the membranes. There's a nylon fine mesh, generally around 10 microns, to protect the membrane and from anything that might protrude through the stainless steel mesh. And then there's a 0.2 micron membrane, which is the actual separation of the pore water from the cell water. So the prototype sampler has been evaluated in the field. Last fall we went to Fort Dix and we inserted 2 4-foot samplers. The site is a chlorinated solvent site. It's got a zone of silty sand separated from a zone of sand by a coarse sand gravel layer, fairly thin, which produces variable results from the wells. The wells are typically 5 to 10 feet screen intervals, and many of them cross through multiple zones, so the low permeability, high permeability zones. And so the intent of installing the peepers here, particularly as we only had developed 2 at the time, was really just to confirm that the samplers were able to be inserted to the depths we desired, in this case somewhere between 20 and 30 feet below the ground surface, that the samplers could be removed, that the pore water would be unaffected by sediment intrusion, and that the samplers would return all the types of data that we anticipated. So this slide has a number of photographs which basically highlight the insertion and retrieval. In the bottom of the slide you can see the fully assembled sampler, and you can make out the stainless steel mesh in the photograph. The upper left photograph is a picture of the geoprobe rod and the sampler being inserted. and on the right is a picture of Haley, the graduate student on this project, who is removing water from the sample cells and putting it into a sample vial. These samplers were inserted between 18 and 26 feet below ground surface, and they were left in the sediment for 3 weeks before retrieval. The whole insertion, including the time to assemble the prototypes, probably took about 3 hours total, which includes the assembly, the pre-coring, the moving the geoprobe to a new location, insertion, and so it's a fairly fast process. Retrieval is even faster. Retrieval and sampling probably took less than an hour. So this slide presents some preliminary data. Keep in mind that the point of this was really just to determine if the sampler could be inserted, could be retrieved, and return data. It was really less about the comparison between the well water and the sample interval, because we only had a 4-foot sample length, and the well screens were 10-foot well screens, and so it's unclear where the well is pulling water. But what you can clearly see is that the samplers returned information on all the measured constituents that we evaluated, which in this case was chloride, sulfate, TCE, Cis, and VC. TCE was below detection in all the samples, including the well samples. But you can see that the samplers return very high resolution data, that the concentration of chlorinated solvents, or Cis-DCE in this case, changes by over 100% across that 4-foot interval. And the well is returning data that's higher than anything we measured, probably indicating that it's pulling from a more contaminated zone than our sample interval was. The same thing is true of chloride. You can see that the chloride changes very rapidly over a very small span, 20 centimeters, changes by a factor of 2 to 3, and that the well water is generally higher, again suggesting that it's pulling water from a different zone. We also successfully were able to measure the compound-specific isotope composition of the Cis-DCE, and we compared it to the well water, and you can see that the sample at 18 feet and at 22 feet are reasonably similar, and that both of them differ slightly from the well water. Actually, the more negative values suggest that the sample intervals that we evaluated are actually undergoing more degradation, or have been processed to a greater extent than the well water. We also evaluated the microbial population. I'm only showing results for one site, but we have similar data for both locations that we inserted samplers. And so the top figure on the left presents the relative populations of reductive dechlorinators with respect to depth. And so in this case, what we did, the analysis actually returns the estimated numbers of each population, and so to make the data a little bit easier to compare, we've normalized that to the total population, but you actually do get numbers of each of these bacterial types. And we also compared that to groundwater. So this is groundwater that we pumped out of a nearby well and sent for analysis, as well as a biotrap that was inserted in the well. And the point of this analysis was not necessarily to compare to the groundwater in this case, but rather to demonstrate that the samplers would have the sensitivity that we needed in order to be able to evaluate the population even at this high resolution, which they most assuredly do. On the bottom graph, this is a different type of analysis that was done. This analysis was done by Microbial Insights, and the bottom graph is the distribution of co-metabolic type processes: methane monooxygenase, toluene dioxygenase, phenyl hydroxylase, all activities that could impact the chlorinated solvent. One thing you can see is that there's quite a bit of variation with depth, and again that just highlights the usefulness of these samplers to be able to see fairly large differences in populations even over small depth distributions. So from the results of our first field test, we learned quite a bit about how the samplers work, and we were able to make some improvements and design a second version prototype. We increased the diameter of the sampler to 2.5 inches. We did this because in general we will probably core before inserting the samplers. And the reason we went to core before the samplers is not that we have to get it in the ground, but we don't want to cause secondary impacts from compression around the sampler. And so by coring we reduce the compaction that occurs. And so the typical geoprobe core is on the outside diameter, the largest diameter is about 2 inches. And so by using a 2.5 inch rod, we're able to keep a very good seal between the sampler and the sediment, but without excessive compaction. We also went to a single row of velocity cells instead of 2 pairs. This was mainly just to get a better seal between the cover plates and the sampler. And then of course we have threads at both ends of the sampler so that these can be stacked to any length you want. And there's really not much of a limitation here. As long as you can drive the rods in, you could stack from 1 to 20 of these sample rods in order to be able to evaluate any depth distribution you will be interested in. We plan on testing these at another field site late summer of 2016, and we're right now in the currently finalizing the site selection. So in conclusion, we think that we have demonstrated the applicability of the sampler. We've certainly verified that it is able to measure all of the parameters that we set out to measure. The sampler is able to be inserted to depths greater than 30 feet. The sample interval could be anywhere from 4 feet to 30 feet, possibly even greater. Deployment time is between 2 and 3 weeks. It's able to measure chlorinated VOC concentrations at 20 centimeter resolution and with a detection limit of around 20 ppb. It's also able to evaluate the typical biogeochemical indicator concentrations at the same sample interval. These include things like nitrate, nitrite, sulfate, iron, DOC. But really there's no limit to what you can look for as long as you have the adequate water to be able to test. And so there is a finite amount of water returned from each depth, and so there's some selection that has to go on. But most of these analyses only use a very small amount of water, which would allow you to look at a number of other species. The cells are also there's 2 cells per each depth, and so you get duplicate analysis for each of these. We were also able to evaluate groundwater velocity. We've developed a very strong relationship between the mass transfer coefficient of a conservative tracer out of the cell and the measured pore velocity. The samplers are able to measure the microbial community and abundance, and as well as the fate of the chlorinated VOCs using CSIA. And so I'd like to acknowledge the funding from CERDIP that is allowing the development of this sampler. I'd also like to acknowledge the hard work of my grad student, Haley Schneider, and my co-PIs, Paul Hatzinger and Paul Custer Van Groo at CBI Federal Services. If you have any questions, I'd be happy to correspond. My email is on the slide as well as my office phone number, and I'd be more than happy to answer any additional questions. Thank you for your time.